which has a particular type of the expression. Some of them actually express high, some of them actually express low. So when you put this your biological classifier circuits inside the cell, if it match with those profile, microRNA profile, it produce a protein and kill the cancer cell. But if it is not, if it doesn't match, it is not going to work, this classifier circuit. So we can see again in this example, I can show you that how you need a design first and then make some biological function similar to the other engineering. Now, this is another example, and a very interesting example here, because people developed a bomb detecting plant. So what does that mean? So if there's a bomb is coming, if there's a bomb nearby, and this bomb is always giving you diffuse some of the chemical molecules, like explosive molecules. So this plant, engineered plant, can actually sense that, and then the green plant turned into white. And that's exactly what I'm trying to show that here. So when in general it is a green, and at the end you can see it is completely white. So it can actually sense a TNT or RDX, and also it is a machine readable. You can see in the machine the colors how they're different. So this is, I just gave 3 different examples of the synthetic biology. If you want to do that, a big part I actually skip, and that big part is the how is it actually affect. I think the 80% of the chemical engineering of the future will be affected by the synthetic biology. But as your institute doesn't have a chemical engineering field, I'm not going into the detail, but I'm going something else. So this is a very, very pathetic news. So this guy in 2005, 2015 it came, and his name is Alankrit. I know this guy personally because he was 1 year senior to my son's class. So when my son is 4 years old, probably he was in nursery or KG, and he's just 1 year ahead of him. So what he had a problem, this guy has basically his heart has a congenital defect. So what he needs a heart transplantation. So what they got at the end, they got a heart from probably from Tamil Nadu, where a 26 years old guy died from a road accident. So got that heart. But the problem is, this Ankit is just a 5 years old kid, and the 26 years old heart cannot actually fit in here, and as a result, this guy actually died. Now I come to this moment that this kind of the system, I mean, this kind of so many fatality and so many pathetic news can only be avoided if we can able to make a whole heart from the patient's cell in vitro, that is meant outside the body. Then we can actually make a perfect size height for Alankrit and actually put that. Now, I would like to emphasize here, this is the situation where if you bring all the doctors of the world together, they cannot do anything, right? If you bring all the resources of the earth together, all the money together, you cannot give life to Alankrit. So this is exactly the era, exactly the responsibility of the scientists, engineers, and the technologists to develop something heart outside the body. Otherwise, this kind of so many Alankrits in our world every day will die. So this is things that what the responsibility of a technologist or engineer or a scientist in this here. So people try to develop this, right? But this is a very difficult thing. So at this moment, what the people actually they do, they use something called the material scaffold. The material scaffold can be made through the 3D printing. I'm sure that as an engineer, you all know about the 3D printing. And that on 3D printing material, they actually put some of the cells. So the cells takes the structure of that organ. So this is a pig's urinary bladder that people actually make outside the body, right? And people try to make a different kind of hard valve, developing different kind of materials, programming materials, giving some scaffold, put the cells and try to do that. But we cannot make a heart at this moment. So our technology is not there. So this is one technology people are looking for in the tissue engineering, developing some materials with scaffold. Now, I'll just go and check that what could be the other way we can take from the science and engineering, which may help in one day to develop a heart. So this is a particular, I mean, if you have studied, I mean, probably you haven't studied this a particular kind of reaction, and it's called a BZ reaction. Okay, and what is that? It needs only 4 different chemicals. You mix them together, okay, and stir it. Okay, and now you just wait. It starts from a kind of reddish violet kind of color, and then you see after 10 seconds it goes to the blue, and again it is going towards the purple, and that violet color, this violet again goes to the blue, purple, and go. So you see this reaction is oscillating, right? So whatever in your chemistry study, whatever studied in your first year or second year or high school chemistry, you will always see there's a reaction A plus B that goes to C, like the acid-base titration, right? There's an endpoint. But here you see there's no endpoint. This reaction is oscillating. That's a very important reaction. I'll tell you that why in a moment. And then if the same reaction you do in a beaker on a petri dish, but this time you don't stir it, it is unstarred. You just keep there. And what you can see there's a very interesting patterns are emerging from this kind of the system. Now the question is, this is a very old chemistry. People died and people do not know what to do with it, right? And people stop there. And today we know that how important this kind of chemistry is. And so you probably know this person, right? Alan Turing is the father of the computer science. Now, what Alan Turing, you all know that developed this universal computing machine, what's called the Turing machine, and that is the fundamentals of the whole computer science. Now, and here I just wrote this apple. Do you know what is the connections between this apple and Alan Turing? Let me tell you. So Alan Turing is basically, I mean, he was a gay person, and that time in that era in UK, this kind of gayism was a criminal offense, right? So what the judge told Alan Turing, either he has to go for a death sentence or he can take an house arrest, and then they should allow the doctors to actually treat him. So because they used to think that gayism is basically a disease. So definitely, other than the death sentence, he chose the house arrest. But he was very depressed. At the very end, he actually committed suicide by injecting a poison inside an apple and then take a bite from that apple. So Steve Jobs gave respect to this father of the computer science by naming his company as an apple and giving this figure a bite taken from by Alan Turing. Now, we know this Alan Turing is the computation, I mean, is the father of the computer science, but he's basically a mathematician. So he developed some other mathematics to explain a certain kind of the pattern formation. So this is something called the developmental biology. What is developmental biology? Now, you remember that, you don't remember, but I'm trying to say that we know that all of our life started from a single cell. When I was or you were in your mother's womb, you started your life from a single cell. That single cell now makes trillions of the cells in our body, and many of the cells are very different. For example, our skin cell is different than the liver cell, our kidney cell is different than our eye cell, and so much. But very interestingly, the software of the cell, that is the genome of the cell, is the same information as in every cell. Either it's a kidney or the skin, all same genetic information are there, same genome. There is no difference in that genome. However, it functions very, very differently. So which basically means from a single cell, when you're starting the life, we are developing a different kind of the pattern. So some of them are going towards a kidney cell, someone is going to make the hand, some of the heart, and so on. So there's a patterning happening. So people do know today that this is a Drosophila embryo. Drosophila is like a fly, nothing else. So if you actually see that gene, this first when this Drosophila embryo differentiating at the different organs, you can see actually the genes first patterning like that. And what this Alan Turing actually developed a theory, mathematical theory, is called the reaction diffusion system. If you take the chemical reaction and connect with the diffusion and nonlinear dynamics, you can actually explain such kind of the patterning. So probably the reaction diffusion system is the part of making heart or some of the or the whole developmental biology. So taking that approach, the synthetic biology, people actually develop a lot of patterning. So if you see this two pattern, colored pattern, this is just bacterial cells, not even the human cells or a Drosophila cells, just the bacteria. So you program the bacteria in such a way, when they grow, they follow this kind of the pattern. Now, here, one of them is a computer simulation, one of them is an experimental behavior. Now, just looking at it, you can see it is very difficult to distinguish that which is in the computation simulation and which is an actual experiment. So you can bring with the help of the synthetic biology, it is exactly coming to this, like the way simulation you design a car, the way in the simulation you design a rocket, and saying you can design the biology in almost at that level. So this is the kind of the patterning of this, right? So people actually increase this number of patterning. Here you can see 1, 2, 3, 4 patterns. Here I think there are 7 or 8 patterns. You can actually develop those kind of patterns, and all these patterns are mathematically predictive from the beginning. So this gives a very good idea to us that what we can do in terms of making heart. So you can start going towards that level. Definitely far behind. Maybe I don't know how much it will take, 25 years or 50 years to make a heart. But this is, again, people try to, because all these things in bacteria, what I have seen, that is happening in the plate. But actually, heart is a 3-dimensional object. So here, people actually develop a 3D pattern. This 3D pattern is not with the material scaffold, before I say is in the tissue engineering. He had just the cell. You give this program inside the cell. So cells are making this 3-dimensional pattern with 2 different genes, red and green, are expressing at various level. So this is very exciting at this moment. Now, I'd say this kind of tissue engineering is much more easy if you do in space. So in space, what is one characteristics, and that is that absence of the gravity. Definitely, that's not an absence of the gravity. It's a microgravity. So that gravity of the Earth and the gravity of the space has a huge difference in terms of its magnitude, and that's called the microgravity. So here, people, as there is no gravity, you can actually develop a 3-dimensional object using the cell in space. So in the two of these things has been done in International Space Station. So this is a liver tissue they actually developed, grow in the space, and here they produced a colon cancer tumor on the space just by giving the cells. The cells actually grow in the 3-dimensional pattern. So this is very exciting, because probably this gives us an idea that the microgravity can assist making those kind of the heart of the 3-dimensional object when you actually connect it with the synthetic biology designed to itself. But space is not stops here. Space is very, I would say, is a frontiers of our future. And what, as an engineering student, you may or may not know that you can actually do a semiconductor processing in space, and that would be advantageous in many, many different ways. So to make a semiconductor, what you do need is a silicon wafer, right? Now, you probably know the silicon is extremely, extremely brittle. So when you make the silicon wafer in the art, you cannot make as a very big silicon wafer. Today, I think the capacity is somewhere around 8 inch. You cannot make more than 8-inch diameter of a single silicon wafer, because if you make it, what happens due to the gravity, this silicon wafer actually breaks by itself. So it's so fragile. So that's actually one problem making the silicon wafer on the art. Another is, you do know that today when you actually do this micro semiconductor processing, when you make a chip, so now every day the chip is coming small, 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 and small. So now if you print something in the chip, so let's say call it as a wire, so the distance between two wire at today's point in 2020, it is 21 nanometer. So 21 nanometer means, it's, if I can want to tell you how to imagine, so you imagine the diameter of your hair. Your diameter of your hair is somewhere around 25 to 50 micrometer. So you imagine that 21 nanometer. Hello, is there any question? I heard something.
No. Carry on.
Okay. Yeah, sure. Yeah. So this 21 nanometer. So compared to your hair's diameter, you can see 21 nanometer is so small. So now Earth's atmosphere is full of dust, right? You have seen all the time, right? So many dust and very small particles. So any small particles, just more than 21 nanometer, if drops on your semiconductor, then the whole semiconductor is gone, because your wear is just the difference between the two wears is 20, the distance is, the gap is 21 nanometer. So what you need, this is an clean room. It's an Intel clean room, you can see here, and everybody is look like they're wearing the PPEs, exactly they wear the PPEs.